And hope you're all doing well. This is Vinayak Pandey from Genius Mind Mentors, and today we are taking up the chapter The Fundamental Unit of Life from class 9th NCERT. Actually, this chapter is very lengthy in the book, so we have provided short notes plus explanations so that we can help students to prepare for the exams. And we are going to cover up many things in this chapter like plant cell, animal cell, multicellular organisms, unicellular organisms, cell organelles, cell division, and etc. Hope you'll all learn and like. Now we are going to have a look on the timeline related to cell. These are the particular events that have helped us understand cell better. So in 1665, Robert Hooke discovered the cells. In 1674, Leeuwenhoek discovered free-living cells in pond water. In 1831, Robert Brown discovered nucleus in cell. In 1839, Purkinje coined the term pleuroplasm for the fluid substance of the cell. Today it is known as cytoplasm. In 1839, Schleiden and Schwann presented the cell theory. In 1855, Virchow expanded the cell theory, stating that the new cell arises from the existing cell. So there are two types of cells, which is plant cell and animal cell. Now we are going to read or study about the cell theory that was presented by Schleiden and Schwann and later expanded by Virchow. So first point is all living organisms are composed of cell. That is, each and every organism, whether it's plant, animals, or humans, are made up of cells. All cells rise from the pre-existing cells. Later we'll get to know about prokaryotic and eukaryotic cells, then we'll understand the pre-existing cell structure. Then the function of an organism is the outcome of combined activities of the cell. For example, if I am breathing, so there are various cells, various muscle cells which are helping me to breathe. So the breathing is the function which is the outcome of combined activities of my cells. There are various exceptions also. We have stated only two. Viruses are non-cellular organisms, and bacteria and blue-green algae are not true cells. Now we'll have a look on types of organisms. There are two types of organisms on the basis of number of cells. First, unicellular organisms. So as the name says, uni means single. So the organisms made of single cell are unicellular organisms. Example, amoeba. Then we have multicellular organisms. Multi means many. So organisms made of more than one cell are known as organisms multicellular. So example, plants, animals, humans, etc. Now we are going to study about the structure of the cell. So there are three features almost in every cell. First, nucleus. Nucleus is also known as the mind of the cell. Second, cytoplasm is the jelly-like substance as you can see in the image. And plasma membrane is the outer covering of the cell, which is also known as cell membrane. Now let's understand plasma membrane in detail. Plasma membrane is also known as selectively permeable membrane, outermost covering of the cell that separates the contents of the cell from its external environment. Plasma membrane allows the entry and exit of some materials in and out of the cell. So as we have seen that plasma membrane is the outer covering boundary of the cell. So the organisms that want to enter the plasma membrane want to have it permit. So the plasma membrane allows the entry and exit of materials inside and outside of the cell, prevents movement of some other materials, and substances like carbon dioxide and oxygen move across plasma membrane through diffusion. Diffusion is a process which is change of any state of matter into another state of matter. And plasma membrane is present in both plant and animal cells. Then we are going to study about osmosis and types of solutions. Osmosis is the spontaneous movement of a liquid from a region of higher concentration to a region of lower concentration. Concentration is the number of solute present in a solvent. Dilute, we can say. So first of all, there are types of solutions. We have to study three types of solutions. First, hypotonic. Hypotonic solution is the solution in which the medium surrounding the cell has higher concentration. So the cell will gain water and cell will contract. As we can see in the image that it is showing that the cell has the lower concentration. And we have studied that from the region of higher concentration, the water is coming to lower concentration. So the cell will gain water and it will swell. So the best example of this hypotonic solution is raisin. If you put raisin in water for overnight, it will swell, it will gain water because of hypotonic solution. The medium surrounding the cell has higher concentration. Then we'll study hypertonic solution, in which the cell will lose water because the medium surrounding the cell has lower concentration. Look, lower concentration than the cell. Cell will lose water. As we can see that in this example, the cell is throwing out the water. Why? Because the cell has higher concentration than the surroundings, so it will lose water. The best example is if you put a potato in water plus salt. If you add some salt in the water and put a peeled potato in it, the potato will shrink in overnight. And then we have isotonic solution. Medium surrounding the cell has equal concentration as the cell. No change is seen. So if you just put a peeled potato in just water only, no change is seen because both the surrounding and the cell have equal concentration. That's it. Now we are going to understand the structure of cell wall, which is only present in plant cells. Let me make a diagram of so for better understanding. This is the nucleus. This is the cytoplasm. This is the nucleus. This layer is cytoplasm, and this outer covering is plasma membrane. As we have studied, this is the basic structure of a cell. But in plant cell, we do have one more outer covering, rigid outer covering, which is known as cell wall. So what is it written over here? It's a rigid outer covering which lies after plasma membrane. It is mainly composed of cellulose, permits the cells of plant, fungi and bacteria to withstand very dilute external media without bursting. So we'll see that this is the cell wall and this is the plasma membrane. This is the plasma membrane and this is the cell wall. So the plasma membrane is exerting pressure on the cell wall, and this cell wall exerts the equal pressure against the swollen cell. Because of their walls, such cells can withstand much greater changes in the surrounding to interact. And here we come across a term plasmolysis, losing or gaining of water of the cells, which results to shrinkage or contraction. This process is known as plasmolysis. Now we are going to study about nucleus. Nucleus is the mind of the cell. It has a double layered covering called nuclear envelope. It contains chromatin material which changes into chromosomes, which is also visible at rod shaped structures. It has a jelly like substance called nucleoplasm. Nucleus plays a central role in cellular reproduction, and nucleus also has a brain, which is known as nucleolus. Nucleus determines the way the cell will develop and what form it will exhibit at maturity by directing the chemical activities. Now we come across a term called deoxyribonucleic acid, which is also known as DNA. The chromosomes contain information for inheritance of characters from parents to next generation in form of DNA. Chromosomes are composed of DNA and proteins. DNA molecules contain information important for cell construction. And functional segments are called genes, genetic material. So we have heard people say that I have my parents' genes. You have got your mother's eyes. So this is all because of DNA, which has the inheritance information from the parents to next generation. In a cell not divided, DNA is in form of chromatin nets. We can see here in this image, these are called chromatin nets. These are net-like structures which convert into chromosomes later on. Now we'll study prokaryotic and eukaryotic cells. So prokaryotic cells are the cells without nuclear membrane. Pro means primary and karyote means nucleus. So organisms with cells without nuclear membrane is known as prokaryotic cells. They also lack most of the cell organelles. We'll study about cell organelles later on. Then eukaryotic cells, organisms with cells with nuclear envelope, almost every organelle is present. So as you can see in the diagram, there is nuclear envelope is present over here, but nuclear envelope is not present in prokaryotic cell. And as I told you that eukaryotic cells arise from prokaryotic cells. And here we come across a term called endocytosis, engulfment of food in unicellular organisms. For example, amoeba with pseudopodia. As we have studied in class 7 that amoeba has pseudopodia, which is the false feet of the amoeba. So amoeba engulfs its food. It feels it and engulfs it. So this process is known as endocytosis. Now we'll study about cytoplasm and cell organelles. Cytoplasm is the fluid substance inside the plasma membrane. It contains many cell organelles. It protects the nucleus. So there are two functions of cytoplasm. It contains cell organelles and it protects the nucleus. Now we'll study about cell organelles. So these cell organelles are the functional parts of the plant cell or animal cell. They perform many functions of the cell. So if I say that there are cell organelles in cytoplasm, so the function carried out in the cell is the combined work of cell organelles. So some cell organelles we are going to study are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. We are going to study about these cell organelles turn by turn. Let us see about the endoplasmic reticulum. Endoplasmic reticulum is also known as ER. It is the short form of endoplasmic reticulum. Endoplasmic reticulum is always near to the nucleus. You can see here. Now, endoplasmic reticulum is a large number of membrane bound tubes and sheets. Looks like long tubules or round or oblong bags. It's of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, why is rough endoplasmic reticulum rough and why is smooth endoplasmic reticulum smooth? We'll study that. As you can see over here, this pink substance is rough endoplasmic reticulum, and there are various things attached to the base of the rough endoplasmic reticulum which are known as ribosomes. So it has a rough surface because it has particles called ribosomes attached to it. Ribosomes manufacture proteins for the cell, then the proteins are sent to various places for work. So these are the functions of rough endoplasmic reticulum. Now we'll study about smooth endoplasmic reticulum. Smooth endoplasmic reticulum helps in manufacturing of fat molecules or lipids. Proteins and lipids help in manufacturing of plasma membrane. So the plasma membrane, the outermost covering of a cell in animal cell, is formed of fat molecules and lipids and proteins. So SER helps to make them. So this process is known as membrane biogenesis. So we can say what is membrane biogenesis? Membrane biogenesis is the process by which endoplasmic reticulum form the plasma membrane through proteins and lipids. Some of these act as enzymes and hormones. So the main function of ER is because it serves as channels for transport of proteins between cytoplasm and nucleus. It also serves as cytoplasmic framework and it detoxifies many drugs and poisons. Now we'll study Golgi apparatus. Golgi apparatus is the system of membrane-bound vesicles arranged approximately parallel to each other in sacs called cisterns. So as you can see in the photo, these are approximately parallel to each other. These are the sacs and these are cisternae. Now, material synthesized near the ER is packed and dispatched to various targets inside and outside the cell. So, the proteins, lipids, and fat molecules which are produced by the ER is packed in the Golgi apparatus and sent to various parts inside and outside. Now, functions are storage modifications, packing products in vesicles. Complex sugars are transformed into simple, and it is also involved in the production of lysosomes. Now we're going to study lysosomes. Lysosomes are membrane bound sacs filled with digestive enzymes. So, digestive enzymes means that these are the digestive systems of the cell. Now, they are waste disposal system or digestive system of a cell. They digest foreign material as well as the worn out organelles. So, they also digest their own friends, their own organelles, which are worn out or of no use. They have powerful digestive enzymes capable of breaking down organic matter. Now, this is the statement. They are called suicide bags. Why? Because when there is any disturbance in the metabolism, the lysosomes burst out and their powerful enzymes digest their own cells. So that's why lysosomes are known as suicide bags. We are going to have a look on mitochondria, which is the most important cell organelle. It is known as the powerhouse of the cell. We'll study why. So, firstly, we'll have a look that mitochondria has two coverings: outer membrane and inner membrane. Outer membrane is porous and inner membrane is deeply folded. As you can see over here, these are folds. Now, these folds increase the surface area for adenosine triphosphate (ATP), which is a energy currency of the cell. It generates chemical reactions. So we can see there that ATP is known as the energy currency of the cell. Now we have one important statement over here, that mitochondria have their own DNA and ribosomes. Now we are going to have a look on plastids. Plastids are present only in plant cells. These are the color giving organelles. These give the color to the plant. These are of two types: leucoplasts, which are white or of no color, and chromoplasts, colored. So, chromoplasts contain the pigment for chlorophyll, which is important for photosynthesis and transpiration. So, chromoplasts contain the pigment chlorophyll known as chloroplasts. Chloroplasts are important for photosynthesis. Chloroplasts also contain various yellow or orange pigments. We may have seen purple colored leaves of plants. So, these are chloroplasts which give the purple color to the leaves. And if there is a question, why are leucoplasts white? So leucoplasts are primarily organelles in which materials such as starch, oil, and protein granules are stored. Chloroplasts consist of numerous membrane layers embedded in a material called stroma. Look, these are the chloroplasts, which consist of numerous membrane layers embedded in a material called stoma. Plastids also have their own DNA and ribosomes like mitochondria. So we have studied two cell organelles which have their own ribosomes and DNA, which is mitochondria and plastids. Now we are going to study vacuoles. Vacuoles are the storage sacs for solid and liquid contents, generally small sized or absent in animal cells. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. Look, this is so big that it is covering a large number of cell volume. In plant cell, they are full of cell sap and provide turgidity and rigidity to the cell. Vacuoles store amino acids, sugars, various organic acids, and some proteins. In unicellular organism, food vacuole is present, like amoeba. Amoeba has only one food vacuole. Sometimes vacuoles help in expelling waste and water from the cell. Again, we can see the example of amoeba. We have finished of the cell organelles, and this is the NCRT given image of the plant cell and animal cell. So we have discussed rough endoplasmic reticulum. We have discussed chloroplast, we have discussed nucleus, Golgi, nuclear envelope, we have discussed in the part of nucleus, plasma membrane, cell wall, mitochondria, ribosomes we have discussed, chloroplast is discussed, cytoplasm is discussed, smooth endoplasmic reticulum is discussed, lysosome is discussed. The in animal cell also all the things are discussed. So we have covered the portion of cell and cell organelles. Now we are going to study cell division, another major important thing. Cell division is the process by which new cells are made. It is of two types. First, mitosis. Most of the cells divide. In this process, every cell is dividing. Each cell is called mother cell. So this is the mother cell. This is the first mother cell. This is the second mother cell. This is the third mother cell. And from these mother cells only, these daughter cells are arising. So each cell is called mother cell and daughter cells have same number of chromosomes as mother cell. Look, these are four number of chromosomes. These are the daughter cells. They also have four number of chromosomes. And these are the daughter cells which have two number of chromosomes. In total, they also have 8 chromosomes if we count that. So this is mitosis. Now it helps in growth and repair. Mainly two cells are formed. Okay, so this was the first cell out of which two cells came, and out of this cell two cells came. So this is mitosis, process of mitosis, which helps in growth and repair. Now let's study another reproductive process, which is meiosis. Meiosis is when fertilization gives rise to offspring cells divided by new process called meiosis. In this, four new cells are formed, and only this is the mother cell. These are all daughter cells. And four new cells are formed. So basically, when the meiosis stops, four new cells are formed. The four new cells have half the number of chromosomes of mother cell. So this has four cells, four chromosomes. This has two. This has only one pair of chromosome. So 4 to 1. 4 to 1 is the process that we can see that the four new cells have half the number of chromosomes of mother cell, and it helps in reproduction. And here we finish our chapter. Thank you everyone for watching the video. Hope you liked, subscribed and shared the video, and we'll be back again with a new video. I hope you found this informative.